Okay, good afternoon, everybody, and very welcome to everybody who's attending our workshop today. So we're very fortunate to have three very distinguished speakers and customers today. We'll be sharing more about the research they do and how the novel enzyme solutions that Kapa engineers has enabled and benefited their research. My name is Marijke Appel. I'm the technical director at Kapa Biosystems. And then we also have Shauneen here. He's a product manager. Lindsay Wiltz at the back. And then we also have Charles Joseph here. So please feel free after the workshop to hang around if you have any questions or want to discuss anything. We'll be available for the rest of the afternoon. So just a little bit about our company. We've been around for almost 10 years now. Kapa Biosystems was originally founded by four entrepreneurs and scientists from the US in Cape Town in South Africa. So we have a kind of interesting history. Our main R&D and production facility is based in Cape Town. And then we have our world headquarters just north of Boston in Massachusetts, which is where I'm originally from, Cape Town, but now based in the US. So we have about 150 people across the two facilities worldwide, probably growing to about 200 by the end of this year. We distribute our products worldwide, so in North and South America, we service from our Boston location, and then everything else from the Cape Town location. We have a very proud history of quality, so we manufacture all our own products in Cape Town. Our company has been ISO certified for many years. Even when we were a 20-person company, we already did that. And we're in the process of transitioning to ISO 13485, which is allows us to just better service our clinical and pharmaceutical customers in the future. And so, as I said, we're in Wilmington in Massachusetts, just north of Boston, and in Cape Town. And then we've just also recently, on January 1st this year, we are now distributing directly in the UK as well. So we have a UK location. So just a little bit about our technology. This is something that I like to talk about a lot, but I don't want to take any time away from our speakers today. So again, if you like to discuss more and are interested in we can discuss afterwards. But just very briefly, we don't do rational design. I mean, as a small company with very limited startup funding, it would take us a very, very long time if we needed to engineer enzymes by rational design. So the strategy that we follow is a much more powerful one. It's called directed evolution. And essentially, we just start with a full-length, genes. Say, for instance, the gene that encodes Taq polymerase. We use error-prone PCR and a combination of mutagenesis and recombination methods to make full-length gene libraries. So these are different to the next-gen sequencing libraries. They encode, entire full-length genes. And the first challenge of this approach is to try and make really, really large libraries, because we are going to be looking for things that are randomly that have a random advantage in an application that we're interested in. You have to generate really, really large sequence libraries. It's almost like I think the odds of finding something that is fundamentally different is pretty much the same odds as winning the Powerball. So you can imagine. And so the first, part of our technology development was just trying to figure out how to make really, really large gene libraries. And so the second challenge is how do we find the interesting things in these really large gene libraries? And again, as a small company, if we had to individualize clones, we would be screening forever before we find something. So instead, we use some of the technologies that's used in next-gen sequencing to allow massively parallel selection in our case. So once we have the gene library in E. coli, we express the protein a little bit. Each E. coli cell then contains a little bit of mutant polymerase plus the gene that encodes the polymerase. So pretty much every E. coli still contains a functional Taq, but they're all slightly different. And then we emulsify the E. coli cells in pretty much the same sort of emulsion that's used in 454 and ion torrent sequencing. And during the emulsification process, we create a PCR reaction. So you already have the polymerase, which is expressed by the E. coli. You have the template, which is the plasmid that encodes the mutant polymerase. And then we design a PCR assay that essentially becomes the selection pressure. So in this case, we want to make it an enzyme that's tolerant to cybergreen, and we just add a lot of cybergreen to that reaction to an extent that's higher than what the wild type can handle. And then under those conditions, we get every mutant polymerase to copy its own gene. And so obviously it follows those ones that have mutations that are just, by chance advantageous. In that setup, those mutant polymerases will copy their own gene many, many times, whereas the other ones will basically just disappear from the gene pool. And so at the end of the first round of selection, we just clone all the full-length genes. We don't have to individualize, never look at them. We go through a second round of that, and then you make the pressure a little higher. And so where you'd start maybe with 10 to the 8th clones in the first round, you'll maybe have 10,000 of those that survive the first round of selection. We'll kind of drill that down to about 100 in the second round, and eventually we get them down to a manageable number. And then that's where the real product development starts. That's where we start figuring out how to optimally express them, how to formulate buffers, and the different applications. And so it's a wonderful system because it allows us to do a whole lot of things. It allows us to not have to be that clever. So we don't have to rationally understand the basis of everything we do. For instance, we don't have to know which amino acids are associated with thermostability. We can just heat things up a lot when we do the selection pressure. We don't have to know what's the mechanism of inhibition by whole blood. We just put a bunch of blood in the reaction, and then we find these mutants that can deal with the situation. So it allows us to address things that are difficult to design rationally. The system always finds more than one solution to the problem. So when we go into product development, we always have more than one candidate. And the really great thing about the system is there's a lot of background advantages. So irrespective of what we're trying to achieve, because all our enzymes are now taken from having evolved in nature to copy genomes to having evolved to do PCR. So every single clone that survives the system has an improved ability to do PCR. And so all our enzymes end up being faster, more robust, and has the ability to amplify different things with similar efficiency. And so over the years, we started sort of in 2006. We've used the system. For instance, we've made a very fast polymerase. We made the world's fastest polymerase. We call it 2G fast. Instead of using a minute per kilobase extension time, you can use a second per kilobase. It just makes screening a lot easier, and you don't lose sensitivity in the process. Its sort of counterpart is called 2G robust. It deals with ethanol, salts, urea, all kinds of common PCR inhibitors. Really great for amplifying GC-rich and AT-rich stuff. We made an enzyme that works in whole blood. We made an enzyme that works with crude plant extracts. In the qPCR space, we made this enzyme called Kapa Cyberfast, which is really tolerant to cybergreen. So in addition to having a really robust enzyme with low amplification bias, we can also put a lot more cybergreen in the reactions to increase the sensitivity of the reaction and the specificity in the end. And it's sort of our cyber enzyme, and just the fact that things are really different and enabling is reflected by our publication record. So to date, we have almost 3,300 publications that reference Kapa products. Since 2008, which is quite impressive, and we're very proud of that. But the two enzymes that kind of got us in the NGS space was our cyberfast enzyme, and then an enzyme called Kappa Hi-Fi, which has been shown to be the optimal enzyme for NGS library amplification because of its ability to amplify different things with similar efficiency. And that kind of got us in the NGS space. In the last few years, we've been very focused on really optimizing library construction, and as we're doing with our enzymes with the rest of our kits, to just get the most out of whatever available sample you have, trying to do things faster, more convenient, and keep on kind of pushing the limits of performance. And that's pretty much what we've been about. So we're very fortunate to have three of our customers here today to speak about their research and just indicate how some of our enzymes have been enabling and supportive of that. So thank you very much for speaking with us today.